and welcome to the 9th episode of the BioLogic podcast. In the first 2 episodes of this series, I discussed the physiology of the cell, its organelles, its cytoskeleton, its transport mechanisms, that kind of thing. In this episode, I'm still going to be discussing the cell, but I'll be moving beyond the basic physiology, and I'll be discussing energy acquisition and manipulation, so how the cell gets energy and how it uses the energy to keep itself alive. After all, the complex physiological processes of the cell, they take a lot of energy to maintain. The things that I discussed in the earlier episodes, like active transport and the flexing of myosin proteins, this all requires energy. Enzymes are proteins that enable the cells to capture this energy and manipulate it or control it in some way to perform a favorable reaction. So let me start with the basics, with the first law of thermodynamics and some basic principles of chemistry. Primarily, what energy is and how it works on a molecular level. You're probably familiar with kinetic energy. The energy like momentum or acceleration of a moving particle is kinetic energy, and it's often called E sub k. Potential energy, or E sub p on the other hand, is the energy of a static object that's in a particular position. So a car at rest on top of a hill has a high potential energy. It has the potential to fall somewhere and release a lot of energy somewhere else in a lower energy position, like a lower ground state. And this is why a car at the bottom of the hill has a low potential energy, because it's at the bottom of the hill. There's nowhere for it to fall. Similarly, a molecule whose configuration or structure is under some kind of torque or pressure, it'll have a potential energy. And this energy can be released or captured by releasing or easing the torque. So if a muscle is twisted in some position, and say this twisting induces a lot of torque on the molecule structure, if you can release that torque somehow, the springing back of the molecule into a lower energy state, a more relaxed state, that springing energy can be captured and used for some useful function. For example, the myosin heads in muscle fibers, they bind with phosphate groups, and this induces a powerful kicking motion in the molecular structure. And so billions of myosin heads, billions of these little molecules, all repeatedly kicking outwards and pulling the actin filaments along with them. This kicking motion collectively moves your muscles, and it allows you to walk, blink, stand, and everything else. The total energy of a whole molecule and all of its parts, including its role in heat, pressure, and volume of its solution, is called the molecule's enthalpy. Let's say you're on a water slide at a theme park. Just humor me with this example for a moment. When you sit on the top of the slide, slowly inching forward, down the slope, you have a high potential energy. You're not going very fast, so you don't really have any kinetic energy, but you're at the top of the slide. You have a long way to fall, so your potential energy is very high. When you begin to slip and accelerate down the slide, your potential energy then turns into kinetic energy. Let's say that, unfortunately, there's a little kid, and he's playing dangerously close to the mouth of the water slide. When you come out of the slide, you come out with a high kinetic energy, and you smash into the little kid with some amount of force. So if the slide had kind of a shallow slope, and you really didn't get going that fast, you wouldn't smash into the kid with that much force. He'd probably be fine. He'd look at you and tell you to watch where you're going. But what if the slide was one of those really steep ones? Like what if it had a steep drop, or it had like a spiral section, and that accelerated you really fast? In this case, you would have a high kinetic energy. So when you smash into the kid, all of that kinetic energy gets turned into mechanical energy of the physical impact. So the kid is hit with more force, and instead of making a snarky comment, he'd probably start crying and go get his mom. On a smaller level, the atomic particles and ions and molecules in biological creatures all float around in their watery world, and they smash into one another, and they convert their kinetic energy into different forms, like mechanical or thermal energy. If a solution full of particles is heated up, the particles move faster, they vibrate more, and they collide with more force because they have more kinetic energy. Thermal energy is being directly added to the solution, and this excites all the particles within it, and it increases their kinetic energy. Differences in concentration can be used to create concentration gradients, and this will induce the directed flow of certain particles. And like a hydroelectric dam, enzymes and other proteins can be set up to harvest the energy from this atomic flow. Consider how electrons have a negative charge, and also consider how negative and positive charges attract one another. When electrons are near other electrons and far from the positively charged protons in the nucleus, they have a high potential energy. The negatively charged electrons repulse one another, and they get attracted by the positive charges of other atomic nuclei. An atomic electron is also attracted to the positive charges in its own nucleus. An electron in the valence shell, that is, in the outer shell, is far from its nucleus. Like the water at the top of a waterfall, the valence electron has a high potential energy. When the electron falls down the orbital shells, it comes closer to the nucleus, and it moves through the electron clouds faster. Its potential energy is being converted into kinetic energy, just like the water in the waterfall accelerating down to the surface of the Earth. This shows how energy is changed from one form into another. It's not destroyed, demonstrating the conservation of energy and the first law of thermodynamics. When molecules react, their enthalpies can change as a result of the chemical modifications made during the reaction. This change in enthalpy is typically denoted as delta H, where delta is the symbol for quantity of change, and H is the symbol for enthalpy. If molecules react in such a way that lowers their enthalpies, heat energy has been expelled. When heat is expelled, or released, the reaction is exothermic. So if you touch something and it's hot, it's giving off heat, it's exothermic. The delta H is negative because heat has been released from the molecules and their enthalpies are lowered. On the other hand, when heat is absorbed in a reaction, the molecules involved increase their enthalpy by literally absorbing the heat energy. These kinds of reactions are endothermic. The delta H is positive in endothermic reactions because the enthalpy of the molecules increases. Besides enthalpy, there's also entropy. Entropy is represented by a delta S, and this means the amount of chaos or disorder that there is in a system. When molecules react and the disorder increases, the entropy increases as well. When molecules react and the disorder decreases, the entropy decreases. In a closed system, entropy always increases, and the available energy always decreases. This is the second law of thermodynamics. Both of these qualities, the entropy and the enthalpy of a reaction, are used to calculate the spontaneity of the reaction. The spontaneity of a reaction, or that is to say if it happens spontaneously or not, depends on the amount of energy required to get the chemical reaction started. So if a chemical reaction needs a lot of energy to be put into it to initiate, maybe once you initiate the chemical reaction, it can kind of propel itself, propagate itself, but it just takes a huge energy investment to get started. If that's the case, that reaction is pretty non-spontaneous. It's not going to just randomly happen. It's going to need some kind of big energy input to happen. On the other hand, some reactions may not require any energy at all to get started. In this case, they can be called spontaneous. They happen basically the moment the reactants come in contact with one another. They don't require a large energy investment like other reactions. The spontaneity of a reaction doesn't necessarily say anything about its rate. The rate of a reaction depends on many factors. I'm not going to discuss them now because it's pretty complicated. But consider like a chemical explosive, something that reacts explosively. If you have a solid piece of sodium and you throw it into a lake, that sodium is going to react spontaneously and violently with the water, and it's going to make a big, huge explosion. It's going to make multiple explosions as more and more sodium gets exposed to the water. On the other hand, some spontaneous reactions they'll occur spontaneously, but they react slowly, and they take a long time to reach saturation or the depletion of the reactant. Spontaneous chemical reactions reduce the amount of free energy in a closed system, creating disorder. Non-spontaneous reactions create order, but they often require energy to get started. They require that big energy investment. The equation used to calculate the spontaneity of a reaction produces the Gibbs free energy change, a variable represented by delta G. This equation is delta H minus T times delta S. To break it down, the equation is delta H, or the change in enthalpy of the reactants, minus the product of the temperature in Kelvin multiplied by the delta S, or the change in entropy. Recall how I described a solution receiving heat energy, and how this caused the particles in the solution to move faster. It increased their kinetic energy and made them strike and bounce off one another more often. When the temperature is high, the molecules are moving fast, and this makes the amount of disorder relatively high. Because of this higher disorder at higher temperatures, entropy plays a larger role in the total free energy change of the reaction. This is why the temperature variable is multiplied by the change in entropy. When the Gibbs free energy change, or delta G, is a negative value for a given reaction, that reaction is exergonic. Exergonic reactions will occur spontaneously. On the other hand, non-spontaneous reactions have a positive delta G value. These non-spontaneous reactions are called endergonic reactions. Because they're non-spontaneous, endergonic reactions require an energy investment or some kind of energy input, or a reduced activation energy in order to get started. Enzymes are life's solution to this problem. Enzymes are catalytic proteins, and they lower the activation energy of a particular reaction. Many enzymes will exploit the energy released by exergonic reactions and use this to power the endergonic reactions of other chemicals. When an exergonic reaction fuels an endergonic reaction in this way, it's known as energetic coupling. Energetic coupling is crucial for all life to exist. Many of the fundamental chemical processes that keep our cells alive all work on a coupled system. Sometimes, there's many systems that are all coupled together and they all interact in a very complex chemical ecosystem. When two or more molecules react, they're basically colliding with one another at some location within the solution. The molecules collide with each other at a particular orientation where the electrons involved in the reaction are brought into close proximity. If there's enough free energy in the system, a collision at the right orientation will lead to a reaction. When the concentration of the reactants is high, the number of collisions between the reactants is also high. Imagine if you're playing bumper cars, but there's only one other person playing. If both you and the other driver just aimlessly bounce around, chances are that you won't hit each other that often. You'll spend most of your time moving through the fenced-off driving area, bouncing off the edges. You might pass each other a couple times, you might have a few near misses, but you won't hit each other that often. But what if there was a dozen people driving bumper cars with you? With a higher concentration of players, you'd be crashing into people every 2 or 3 seconds, on all sides. Just like molecules, a higher concentration of reactants in a solution will increase the number of times that they can collide in a suitable orientation and initiate a reaction. Similarly, the number of collisions will increase if the temperature also increases. Go back to the scenario where it's just you and one other person in bumper cars. You wouldn't bump into one another very often, but what if you increased your speed? Instead of the awkward, kind of jerky swerving of a normal bumper car, you're both now in a supercharged bumper car, and both of you drive really fast. So assuming that no one flips over or breaks their car, you should be bumping into each other a lot more often, simply because you're covering more ground in the same amount of time. When you increase the temperature of a solution, you're adding thermal energy to all of the particles in it, and you're exciting them, making them vibrate and move faster. Now, it shouldn't be hard to understand why you can increase the rate of a reaction by increasing the temperature, the reactant concentration, or both. As a student of biology, it's very important to understand things called oxidation-reduction reactions, or redox reactions. If you're just casually interested in biology, it's obviously less vital that you understand redox reactions, although it'll help you understand a lot of the more complicated biological processes, that rely on them. In an oxidation-reduction reaction, one reactant will take one or more electrons from another reactant. The reactant that takes the electrons is called the oxidizer, because it oxidizes the other reactant. The oxidized reactant is the one that loses electrons. The oxidizer, the reactant that took the electrons, is reduced. By taking the electrons, the reactant also takes on the negative charges of those electrons. The charge of the reactant molecule has thus been reduced because it's taken on negative charges. Just as the reduced molecule is called the oxidizer, the oxidized molecule is called the reducer, or the reducing agent. It's a two-sided relationship. Redox reactions can also be understood from the perspective of the electrons themselves, as they're transferred and shuttled between molecules. Electrons are often, but not always, shuttled on protons, or hydrogen atoms, which means that a traded electron with its negative charge is often accompanied with a proton and its positive charge. The electron donor releases the proton-bound electron, and this is received by the electron acceptor. Recall that molecules with lots of carbon-hydrogen bonds, like lipids and carbohydrates, they have a high chemical energy. When an electron acceptor binds to a proton, like in a redox reaction, it can increase the total energy of that molecule. Because this is a two-sided relationship, this also means that the electron donor, the molecule that gave the proton away, it loses potential energy because it released the proton. Consider this example of a very fundamental redox reaction that occurs in all living cells to produce energy. The oxidation of glucose to produce carbon dioxide, water, and energy. So a molecule of glucose contains 6 carbon atoms. And when these carbon atoms are exposed to molecular oxygen, they can be metabolized in a redox reaction for energy. This redox reaction breaks apart the glucose molecule, carbon by carbon, reconstituting the atoms into molecules of carbon dioxide. In the glucose molecule, the carbons share their electrons equally. In carbon dioxide, the electronegativity of the 2 oxygen atoms pulls the electrons away from the central carbon atom. The carbon atom in the carbon dioxide, it's basically oxidized, as it's effectively lost its electrons. Water molecules are also produced in this reaction. In these water molecules, the oxygen atoms are bound to hydrogen atoms instead of other oxygen atoms. The molecular oxygen, it was 2 oxygen atoms bound together. And because it's the same atom, they have the same electronegativity, so they share their electrons equally. But in water, the oxygen is paired with hydrogen, and hydrogen has a weak electronegativity. And so it ends up happening that the oxygen in the water holds its electrons much closer than the hydrogen atoms can. And this means that the oxygen atoms have effectively gained electrons, so they're basically reducing themselves. Their electronegativity attracts electrons to them, and they are reduced. So because the products of this redox reaction, carbon dioxide and water, they all hold their electrons closer to the nucleus than the reactants, which were glucose and molecular oxygen. Because of this, the products have a lower potential energy and a higher entropy than the reactants. Finally, the reaction releases energy as heat, making it exergonic. In earlier episodes, I've referenced a molecule called ATP, or adenosine triphosphate. This small molecule is often used for energy because it contains a chain of 3 phosphate groups, hence the triphosphate. Each phosphate group is composed of a phosphate atom bound to 4 other oxygen atoms. When bound together in a chain, the triphosphate group contains 4 negative charges and a dozen strongly electronegative atoms, all held tightly together in a really small space. Like the electrons kept near other electrons, these tightly bunched up phosphate groups have a very high potential energy. During a hydrolysis reaction, the outside phosphate group is cracked off like someone taking a bite out of a granola bar. The products of this hydrolysis reaction are ADP, or adenosine diphosphate, denoting the 2 remaining phosphate groups, and 1 inorganic phosphate ion. Most importantly, this reaction produces a huge amount of energy from breaking the bond open. Cracking off that last phosphate group releases 7.3 kilocalories per mole of ATP. A kilocalorie is the quantity of energy needed to raise 1 kilogram of water by 1 degree Celsius. Recall that water has a relatively high heat capacity, and it requires a significant energy input in order to heat up. So 1 mole, or 6.022 times 10 to the 23rd molecules, of ATP could then be cracked open to heat up 7.3 kilograms, or almost 16 pounds of water by 1 degree. If you've ever boiled a stovetop full of water, you know that it can take a fair bit of time, and that's only for 3 or 4 pounds of water max, So 16 pounds of water is like 1.5 gallons. 1.5 gallons of water can be warmed up a whole degree Celsius by just a single mole of ATP. That's an incredible amount of energy. Absolutely incredible. Especially since it comes from something so small, the little humble ATP molecule. Besides this amazing release of energy, the ATP hydrolysis reaction is exergonic because its products have a higher entropy than its reactants. There are more products than there are reactants in the first place, which directly increases the amount of disorder, and thus entropy, in the system. The products also have a much lower potential energy than the reactants, meaning the total free energy in the system has been lowered. So now you might be asking, how does the cell actually capture and use this energy released by ATP? Isn't it a bit like trying to bottle an explosion with your hands? Maybe. Kind of. The physics of chemistry aren't quite the same as the physics of our macroscopic Newtonian world. In an explosion, a lot of the energy is released as heat. If the ATP released all of its energy like an explosion, most of it would be lost as heat. Instead, the bond energy released by cracking off the phosphate group is used to smash it onto a new molecule, literally onto the structure of another molecule. The act of a phosphate group being transferred from ATP to a new molecule is called phosphorylation. Upon binding the phosphate group, the new molecule is phosphorylized. This addition of the phosphate group, or this phosphorylation, brings a strong negative charge onto the molecule's structure. This very negatively charged addition can change the conformation of the phosphorylated molecule, increasing the potential energy enough to make them able to react with something. Molecules that must be phosphorylated in this way before they can perform certain functions, they go between two states, an inactive state and an active state. In some molecules, phosphorylation induces conformational changes that make it more reactive. This molecule could be said to be activated. In other molecules, the active state doesn't require a phosphate group. In these cases, phosphorylation will actually cause a conformation change that inactivates the molecule. In this way, phosphorylation can act as a regulatory tool for numerous molecules like enzymatic proteins. Enzymes are a really amazing class of protein molecules. Amazing for many reasons, but primarily because of the breathtaking variety of functions they can perform and the reactions they can catalyze. Without enzymes, life could not function, period. Many of the chemical reactions that life depends on are facilitated by enzymes, and they couldn't happen without them. Enzymes are typically large, flexible proteins, and they possess specific regions in their structure called active sites. These active sites are proximal groups of amino acids with specific R groups that show an affinity for particular molecules. These particular molecules can bind to the active sites, and this causes a change in the enzyme. The change in the enzyme facilitates some reaction or modification between the bound molecules, reaching an unstable transition state between reactants and products. The transition state is unstable because the original bonds are being broken while new bonds are being formed. It's an unstable moment in time for the molecule being modified, for that poor ligand, stuck in the rubbery grip of the enzyme's active site. Depending on how unstable this transition state is, it can be harder for the enzyme to make it happen. In other words, the more unstable the transition state, the higher the effective activation energy. As I've said, molecules bind and react when they collide at particular orientations, and with enough kinetic energy to overcome the activation energy of their reaction. This activation energy, or E sub A, is the energy required to initiate a particular reaction. It usually doesn't take much, if any, energy to keep a reaction going once the initial energy investment is made. But for non-spontaneous reactions, it takes that energy investment, that big energy dump, like a match being lit, to begin the reaction. If two reactants are mixed together in a solution, but they don't have sufficient thermal or kinetic energy to overcome the activation energy, they won't react. So in this way, the activation energy is like a barrier. If there's not enough energy to propel you over the barrier, the reaction can't begin. It just runs into the barrier and stops. So the rate of a given reaction is heavily influenced by its activation energy. If this energy barrier is high, then the reaction is less likely to happen, and the total rate of the reaction will be slower. If the energy barrier is low, then the reaction is more likely to happen, and the rate of the reaction will be faster. In this way, both activation energy and the thermal energy, or the temperature of the particles, influences the rate of any given reaction. Even in a high temperature solution, a reaction may proceed slowly if the activation energy remains high. This is where enzymes come into play. Recall how I described the active site of the enzyme as a group of amino acids in the structure with R groups that have an affinity for the molecules they interact with. When a target molecule binds to the active site, its transition state is stabilized by these R groups. A more stable transition state means a lower activation energy. If you watched episode 3 about proteins, you should remember that amino acid monomers are part of the polypeptide chain. Each amino acid has one of 20 different possible R groups, and these can be nonpolar or polar, they can be acidic or basic, they can be positively charged or negatively charged. Depending on the qualities of the reactant molecules, The active site of the enzyme could have any combination of R group side chains, and these happen to fit any possible number of reactant molecules. These R groups don't just stabilize the molecules with hydrogen bonds and polar affinities and stuff like that. They can also facilitate the transport of various atoms or groups of atoms from one reactant molecule onto another. Enzymes that facilitate redox reactions have basic or acidic R groups in its active site, and this helps move a proton from the donor to the acceptor. Ultimately, the specialized active site allows the enzyme to bind one or more substrate molecules and catalyze some kind of reaction between them. The process of enzymatic catalysis has 3 steps: initiation, the transition state, and termination. In the initiation step, the substrate molecules bind to the active site. The enzyme is able to manipulate the molecules into an optimal orientation for binding. This orientation process is called an induced fit, and so it's kind of like the enzyme is taking 2 different pieces, turning them or twisting them a little bit to get them to fit together. So in the second step, the enzyme handles and processes the unstable transition state of the reactants. In the first step, initiation, the enzyme locked the pieces together, and this allowed them to begin reacting. And so in the second step, the reaction is currently going, and the enzyme has to handle this and kind of keep it stable and keep it going, because if the enzyme fails or breaks apart, the reaction stops. The enzyme's various R groups and conformational changes will help to stabilize the transition state and lower the activation energy. This leads to the third step, termination. After the reactants have been catalyzed, the product molecules have a relatively low affinity for the active site of the enzyme, and they detach. This frees the enzyme's active site, and it allows it to bind new reactant molecules and do the process all over again. Because this 3-step process takes a little bit of time, the enzyme can only process its substrate molecules so fast. If you were to plot the rate of an enzymatic reaction versus the substrate concentration, you would find the reaction rate tapers off asymptotically as the concentration of substrate molecules increases. When there's little substrate, the enzyme can only process it as often as it comes into contact. When there's a lot of substrate, the enzyme can process stuff as fast as it can get its hands on new molecules. So if you add more substrate, the enzymes will be more and more likely to bump into reactant molecules and process them. Eventually, you'll get to a point where adding more substrate doesn't really do anything. The enzymes in the solution are already processing the reactant molecules as fast as they can. Continually increasing the concentration of substrate molecules will lead to a smaller and smaller increase in the rate of the reaction, until you reach a point called saturation, and this is where the rate simply doesn't increase, no matter how much more substrate you dump in. Imagine you're riding a horse. You can have the horse go at a walk, which is just a slow, casual pace. You can make the horse go into a trot, and it'll pick up its pace a little bit, or you can have the horse go into a full-on gallop. And when it's galloping, it's moving very fast. And you can try and get it to go faster, but the horse really will have a top speed that it can go. It's hit speed saturation, if you will. And no matter how much more you encourage it, the horse is not going to go faster. It's going as fast as it can. Enzymes are the same way. If they're saturated with reactants, they can only process the reactants at a maximum speed. Adding more and more reactant, it won't make the reaction go any faster. Now, obviously you wouldn't put in a single enzyme and expect it to process industrial quantities of a substrate in any reasonable time period. You'd put millions or maybe even billions of copies of your enzyme into a solution, and then you'd put in enzymes that helped your enzymes, and enzymes that helped those enzymes, and then enzymes that helped the whole enzyme complex. The point is, enzymes don't work alone. Many enzymes have modular components which are needed for the enzyme to function. Without these components, the enzyme can't do its job. It's in an inactive state, so to speak. These modular molecular attachments are organized into 3 types: prosthetic groups, cofactors, and coenzymes. The prosthetic groups are non-amino acid molecules, so think molecules like sugars or lipids. Cofactors are inorganic ions like zinc or iron, and these engage in reversible interactions with other enzymes. So reversible means these molecules can attach and then detach. Coenzymes are like smaller mini-enzymes, and they're capable of a limited but reversible interaction with larger enzymes. Much like the R groups, these types of molecular enzymatic assistants can help stabilize the transition state or shuttle around atoms, or facilitate the conformational changes in another enzyme, or really perform any role that basically helps stabilize and push along the reaction. Some of these assistant molecules regulate the activity of the enzyme itself. Regulatory molecules are those that bind non-covalently. That is, they bind through like hydrogen bonds or polar interactions or van der Waals forces or something like that with the enzymes. The non-covalent binding of these regulatory molecules to the enzyme can activate or deactivate it. These molecules can also be released from the enzyme without changing its shape or structure, which is why they're called reversible reactions. Regulatory molecules can either bind to the active site or somewhere else on the enzyme. Those that bind to the active site must be similar in size, shape, and polarity to the enzyme's natural substrate molecule. In a phenomenon known as competitive inhibition, the regulatory molecule binds to the active site and basically blocks it, preventing it from binding to the natural substrate molecule. In a phenomenon known as competitive inhibition, the regulatory molecule binds to the active site and basically blocks it, preventing it from binding to the natural substrate molecule. The inhibition is competitive because the substrate molecule and the regulatory molecule are competing to bind to the available active sites. If the regulatory molecule binds to somewhere beside the active site, the interaction is called an allosteric regulation. This binding will cause a conformational change in the enzyme. Some enzymes are activated when an allosteric regulator binds to them, changing their shape into something functional. Alternatively, some enzymes are naturally active, but they become inactivated by the conformational changes caused by binding to an allosteric regulator. Sometimes these regulatory molecules do bond covalently to the enzyme, or they involve modifying the enzyme by breaking some of its covalent bonds. For example, a protease activates the enzyme trypsin by cleaving off a small portion of the enzyme's polypeptide chain. Phosphate ions are a common cofactor in enzymatic activation. The phosphorylation of an enzyme is basically the addition to the enzyme of an ion with a strong negative charge. The negative charges of the phosphate ion react with the polar and charged R groups on the amino acids, and this distorts or bends the enzyme. Just like regulatory molecules, the phosphorylation of an enzyme causes a change in its conformation. This conformational change can activate or inactivate the enzyme. If it inactivates the enzyme, the dephosphorylation will activate the enzyme. Phosphorylation is a reversible reaction. It's important to understand that enzymes are proteins, and just like any other protein, enzymes can be denatured or rendered inoperable by harsh environmental conditions. If the pH of their solution is too high, for example, proteins will denature and cease to function. The same will happen if the enzyme is exposed to an intolerable temperature extreme or a dangerous level of salinity. With this in mind, it should be understood that proteins and enzymes are also specialized for their environments. You and I have proteins that function quite nicely at 98.6 degrees Fahrenheit. Extremophile microbes that live in deep-sea vents have proteins and enzymes that can withstand the heat of their environment, and this environment can sometimes reach over 200 degrees Fahrenheit. So now that you have a pretty decent idea of how individual enzymes work, I want to briefly discuss how groups of different enzymes work together. In a metabolic pathway, an enzyme, let's call it enzyme A, binds to a substrate molecule and catalyzes a reaction to create a product. Another enzyme, enzyme B, treats this product molecule as a substrate and binds to it to catalyze another reaction. Enzyme B creates its own product. Enzyme C, a third enzyme, then binds to that product and catalyzes yet another reaction. Then enzyme D gets involved, and so on and so forth. In this way, a base molecule can be passed through a series of enzymes, each catalyzing a reaction that causes a small modification. So by the time the base molecule reaches the end of the chain, so to speak, the end of the metabolic pathway, it's usually a heavily modified version of its original precursor molecule. At any step along the metabolic pathway, product molecules can be split into groups, and each group diverges down a new branching chemical pathway. Metabolic pathways can also self-regulate themselves through feedback inhibition. In many instances, the product of one enzyme in the pathway will inactivate another enzyme, and this will shut down the whole pathway. This often happens when the products of a pathway have reached saturation and are no longer needed. The products begin binding to and inactivating their own uncle enzymes, shutting down the pathway. It's evolutionarily advantageous to shut down an enzyme near the beginning of the pathway so as to not waste resources half building some kind of product molecule that's just going to be ditched or abandoned anyway. Many of these reactions are catabolic. That is, the chemical pathway generates ATP and carbon molecules by breaking down energy storage molecules like carbohydrates and lipids. Other reactions use this energy and this carbon, and they build other molecules. These building reactions are facilitated by enzymes and anabolic pathways. Anabolic and catabolic pathways are both dual aspects involved in a really important process called cellular respiration. Because you're made of cells, you breathe or respirate for all the cells in your body. You do this by inhaling oxygen and exhaling carbon dioxide and water. But I'll save cellular respiration for the next episode. That's it for this one. I hope you enjoyed listening, and I hope you learned something cool about how the cell uses energy and its enzymes. Thanks for listening. If you enjoyed this episode, hit the like button and subscribe. Share the episode with a teacher, a friend, or a classmate. If you want to support the show, follow us on social media and consider buying something from the official store. Follow us on Twitter. Our username is @BioLogic Podcast. To buy something from the store, go to redbubble.com and search for BioLogic. Or go directly to the store at redbubble.com/people/biologic/shop. Become an official patron of the show. Sign up at patreon.com/biologicpodcast. Your support really helps the show, so if you like it, send us some love. And as always, thanks for listening.